Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you find this deep dive valuable, please do hit that like button and subscribe. It really helps support the Sans Science Experience and allows us to bring you more fascinating insights like this one. Today, we're plunging into a really interesting new preprint. Now, that's a research paper shared before the full formal peer review. It's titled, Semaglutide Slows Epigenetic Aging in People with HIV-Associated Lipohypertrophy, Evidence from a Randomized Controlled Trial. Our mission today, decode what this study reveals about, well, a pretty well-known drug and its surprising potential beyond what we usually use it for. So let's unpack this.
Yeah, it's fascinating because we're talking about semaglutide. Most people know it, or drugs like it. They're GLP-1 receptor agonists.
Right, the ones known for weight loss and diabetes management.
Exactly. They mimic a natural hormone, help regulate blood sugar, appetite, widely recognized for, cardiometabolic health benefits. But this study, it's looking at something else. It's exploring its potential role as a gerotherapeutic.
A gerotherapeutic, so literally a drug targeting aging itself.
Precisely, a drug that could potentially slow down or even reverse biological aging processes. It's a really exciting area of research.
Okay, so let's get into the study itself. How was it set up? And who were the people involved?
Right. So this was actually a post-hoc analysis. The main trial was a 32-week, double-blind, placebo-controlled study.
Post-hoc meaning they looked at the aging markers after the original trial finished.
Exactly. The original trial wasn't specifically designed to measure epigenetic aging, but they had the data. The participants were adults diagnosed with HIV-associated lipohypertrophy. That's a condition where sometimes HIV treatments cause changes in body fat distribution.
Oh.
So they had 45 people getting semaglutide and 39 getting a placebo. And this group is particularly interesting because people living with HIV often show signs of accelerated biological aging.
Ah, I see. And often persistent low-grade inflammation too, right?
That's right. Which makes them, in a way, a really relevant model to study therapies that might protect against aging these potential geroprotectors.
Okay, so this is where it gets really intriguing. The findings on epigenetic aging, they sound pretty dramatic.
They really are quite striking. The study found semaglutide significantly decreased epigenetic age across several different measures, these clocks.
And these clocks, they're based on DNA methylation, like little chemical tags in our DNA.
Exactly. These patterns change as we age, and also in response to environment and lifestyle. They give us an estimate of biological age, which isn't always the same as your chronological age.
So how big were these changes? The numbers mentioned 3.1 years, 4.9 years.
Yeah, pretty substantial. For instance, PCGrimAge, which is strongly linked to mortality risk, it decreased by an average of 3.1 years in the semaglutide group compared to placebo.
Wow, 3.1 years younger in terms of mortality risk.
Uh-huh. And then there's PhenoAge, another key marker of biological age reflecting overall health. That showed a reduction of 4.9 years.
Almost 5 years biologically younger in that sense.
Yes. And another one, DunedinPACE, which actually measures the speed or pace of aging. That indicated about a 9% slower pace of aging in the treated group.
A 9% slower pace. That's quite something. So it suggests a real shift in the underlying aging process.
It definitely suggests that. Well, these aren't guarantees of living longer chronologically. They point towards a significant improvement in biological health markers linked to longevity.
And was this effect localized, or did it seem more widespread across the body?
That's another really interesting part. Looking at the bigger picture, it seemed quite broad. They used these organ-specific clocks too, and semaglutide appeared to slow down aging across 11 different organ systems.
11 systems. Which ones showed the biggest effects?
The most significant benefits were seen in clocks related to the blood, the brain, inflammation, heart, kidney, liver, and metabolic systems. So quite multi-system.
That's really broad. Do we have any idea how it might be doing this? What's the proposed mechanism behind it all?
Well, the researchers connected back to principles of geroscience, the study of aging biology. They point out two key things semaglutide does. It lowers biomarkers associated with inflammation.
Which we know drives a lot of age-related issues.
Exactly. And it also significantly reduces visceral adipose tissue. That's the harmful fat deep inside the belly.
Right.
And the idea is that reducing this fat might directly lessen what they term obesogenic epigenetic memory.
Obesogenic epigenetic memory. Like the body remembering the negative effects of fat tissue through those DNA tags.
Kind of, yes. Those pro-aging signals that seem to come from excess or unhealthy fat tissue. By reducing the fat and inflammation, you might be dampening those signals at a cellular level.
Fascinating. Now, Were there any limitations, any clocks that didn't change, or things we need to keep in mind?
Yes, definitely. Important to be balanced. While many clocks showed these strong effects, not all did. For example, one called the Intrinsic Capacity Epigenetic clock, which is maybe more tied to physical function, didn't show a significant change.
Okay.
And critically, we have to remember this was a post hoc analysis. The study wasn't huge, about 84 people in total, and the duration was 32 weeks.
Right. Relatively short term in the grand scheme of aging.
Exactly. So we definitely need longer, larger trials. We need to see if these effects last, and importantly, if they apply to other groups of people beyond those with HIV-associated lipohypertrophy.
Makes sense. So wrapping this up, what's the big picture takeaway here?
I think the main point is that, as far as we know, this is the first randomized clinical trial evidence showing that a drug in this class, a GLP-1 receptor agonist, can actually modulate these DNA methylation biomarkers of aging.
So real evidence from a controlled trial, not just observational data.
Correct. And it strongly supports the idea that semaglutide might have potential as a genuine gerotherapeutic, something that influences the fundamental processes of aging itself.
So for you, our listener, this study really offers something to think about, a compelling glimpse, maybe, into the future. If a drug many people are already using for other reasons can potentially nudge our biological clock backward, what other existing treatments might hold similar secrets? And, maybe more profoundly, how could this shift our whole approach to health, moving beyond just treating diseases one by one toward actively promoting a longer, healthier lifespan for everyone? Definitely something powerful to consider. And if you enjoyed this deep dive and found it valuable, please, once again, remember to like this video and subscribe to the Sand Science Experience. It helps us immensely. Thanks for joining us.